All right, so it's my pleasure to be the co-chair for this session with my colleague Philip Awadala. We're going to talk about the Team A for IHCC. This is really about the kind of data and kind of interoperability between the cohorts and our plans and thoughts for that part of the IHCC project. Okay, so I'm going to talk about our sort of vision and the kind of challenges that we think are going to be we need to address essentially to get this kind of this large-scale cohort interoperability. Okay, so I mean, Team A. If I was to kind of summarize what our vision is in a short sentence, I guess it's really to deliver the interoperable IT standards and infrastructure to enable all of the kind of research applications, the clinical applications of that you can get from being able to do kind of very large scale cohort studies and be able to find cohorts, to be able to interoperate between them, to be able to carry out, say, federated analysis across them, to be able to access the genetic data across these cohorts. Okay, so I mean, if you think about cohort data and and certainly where it's going, in terms of the large-scale genetic projects and genomic projects that are starting to happen around cohorts with the decrease in the cost of genotyping and sequencing. I mean, we are talking about very large data sizes. Some of the cohorts that are part of IHCC. I mean, it's the nominal minimum kind of entry level was about 100,000 participants. Many of the cohorts are starting to generate genomic data and genetic data. That data is large. The raw data is very large. Even just the genetic variant data is large. On the order of tens of gigabytes for a single sample. We have to acknowledge that the cohorts, they operate in different jurisdictions, different countries, different continents, and there may be restrictions on how that data can be exported across international borders. And of course, a consequence of that is that there's going to be different security requirements for storing and accessing those data sets. And that's it, sort of on the kind of raw data side of it. If you think about the metadata, I mean, we've got heterogeneous phenotypes, we've got different collection protocols with different vocabularies, different data dictionaries, and trying to get some sort of semantic interoperability is probably one of the most significant challenges for cohort interoperability, and we heard a little bit about that this morning from some of the directories. So our current kind of thoughts is that certainly from the first sort of points in this slide, that we think a federated solution is probably going to be the most appropriate approach for getting interoperability with across the IHCC network, and not just within the IHCC set of cohorts, but within when you want to interoperate with other cohort initiatives. That there will always be more than one cohort initiative around the world. We heard from some of them this morning. There are obviously benefits from having a federated model. The metadata and the variables for each one of these cohorts, they can be updated almost at source or maybe at a hub that cohort is participating into. The cohorts can align with other initiatives. We're already seeing that many of the IHCC cohorts are also part of other initiatives as well, and having a federated model means there's flexibility for the cohorts to participate in different initiatives. There are some nice things like not having a single point of failure for the whole net. If you've got a discovery network, you're not going to have a single point of failure for sending out cross-cohort queries. And probably the most important is that we have to acknowledge that we need a flexible data access model, because what type of data can be shared, how it can be shared, how you can access it, that's going to be really heterogeneous across the cohorts. I think probably one of the most significant challenges is this semantic interoperability between the variables and data dictionaries and phenotypes. And I think that really shouldn't be underestimated as probably it's one of the fundamental challenges for being able to even discover cohorts and samples of interest within those cohorts. And obviously, we need to work out, how can we fund the cohort maintainers to be able to actually do this kind of harmonization or this mapping into a sort of a common sort of language for federating queries across for discovery across cohorts. So one of the groups that we heard about this morning from Peter Goodhand. One of the groups I'm quite heavily involved in is the GA4GH, the Global Alliance for Genomics and Health. And we think that we would really want that the Team A activities will be really highly aligned with the GA4GH. This is this group that's developing standards and framing policy for responsible genomic and health data related sharing. They've got quite a lot of, quite well established up and running data work streams and task teams that are quite active and already kind of rolling out standards in a whole bunch of different initiatives. They have got the GA4GH has a matrix model. There are a whole set of real world driver projects. These are projects that are generating data, that are sharing data, that are making it accessible, and they need it. They need technical standards, and there are a whole set of technical work streams in the GA4GH that work on trying to tackle the individual technical challenges around like data discovery, things like data use and researcher IDs. There's a cloud work stream that's trying to get this model of being able to bring your analysis to where the data is located. And obviously the clinical and phenotypic data capture and harmonization. And underpinning that, we have two foundational work streams, the regulatory and ethics work stream and our data security work stream. There are a whole set of projects, real world driver projects. This was the initial list. It's now been expanded again in the last few months. And having these driver projects has been like absolutely key, so that the standards that we develop in GA4GH, they're actually getting rolled out in real world projects to see that they're actually fit for purpose and actually work at scale and in production. Okay, so yeah, one of the if we were to break down the kind of the IT challenges into a set of kind of individual kind of level components that you or challenges that you would try to address. I mean, this is a figure that from a recently funded project that I'm leading, the Seneca project we heard about this morning. It's a Horizon 2020 project, and it's about cohort interoperability and cross-cohort federation and being able to enable federated analysis and clinical applications. And we kind of broke it down into a set of work packages, and they try to address each one of the individual challenges. So I mean, if you break it down, you got cohort level metadata representation. This is the semantic harmonization kind of effort that's needed. It's fundamental for any kind of cross-cohort analysis. Authentication authorization. How do you recognize a researcher? How do they request for access? How do they get authorized? Getting that interoperability. Federated data discovery, that's fundamental to how do you find data sets of interest. And one of the things I'm looking forward to from the RFI presentations is that we need a really set of convincing research applications and clinical applications that you would want to do, that you would want to carry out if you had access to a very large set of cohorts. What are the applications that you would want to actually generate, and what research questions would you want to answer? Okay, so so for the rest of the talk, Philip is going to talk about the sort of aspects of the initial challenge that we're trying to look at is data discovery, and how do you find cohorts? It is one of the fundamental challenges, one of the fundamental barriers for cohort data reuse. It is the first kind of entry point for any researcher or clinician that wants to find cohorts or samples of interest. And we have to think about what are the types of queries that we would want to ask. So I've borrowed this figure here from the GA4GH data discovery work stream, and they've kind of just framed a set of possible queries, and then tried to break them down into a set of kind of individual level components like genetic variants, genes, phenotypes, allele frequencies. What are the types of how would you break these complex queries down into sort of individual chunks that you would then kind of query individually and then combine? Okay, so I'm going to hand over to Philip. He's going to talk about our initial efforts towards a IHCC cohort atlas.
Great. So yeah, so in the emphasis on initial. So we are moving towards collating as much information as we can from programs that have agreed to be part or share information with the IHCC. And that was our starting place. So to some extent, this is what the very wonderful people as part of the IHCC have put together from data and information that was provided by many of the cohorts that are many of you are here. Some of you weren't able to come for this meeting. And we've been trying to cross-reference this with data that we can capture either by doing basic internet searches or things that we've got cataloged in our programs in Canada. And I'll mention the Maelstrom program really quickly. For those who don't know Maelstrom, Maelstrom is an activity that we fund in part out of McGill. Isabelle Fortier is the PI, and she's developed, if you like, a catalog of many, many, many cohorts, and they all vary in terms of size, structure, clinical, most of them are population cohorts. But what we've done is we've tried to identify whether your cohort is in this Maelstrom catalog as a start and see if we can pull data dictionaries from that in helping to try to build this IHCC atlas. With that said, when I went back after the first meeting of this meeting to my team saying, hey, you know, one of the things we've been tasked with is building the IHCC atlas. And somebody said, oh, well, we've already started that. You signed off on that in 2015. And I was like, oh, this is great. Of course I remember doing that. And to some extent, so this is the project. So we supported this activity. And generally, it's called the Cross-Cohort Harmonization Project. It's a Canadian activity that we supported as part of the National Canadian Cohort, where I'm the National Scientific Director. And this was an activity, and one of the reasons I thought this was really cool at the time, I think, was because I wanted to know how well harmonized our variables could be in the Canadian cohort with many other cohorts, right? So this is our start, and this is where we're starting with the IHCC activity as well. So you can see some of these cohorts, many of these cohorts should be recognizable to you. And many of them are already part of the IHCC as well. So there are at least 9 cohorts, and these are some of the larger cohorts that have agreed to share information with the IHCC. So this includes almost 2.4 million participants. So as I said, I'll just do a shout out for the Canadian cohort because I'm here. That's the Canadian Partnership for Tomorrow Projects. That's 333,000 participants. Constance, I think, is going to be close to 300,000. UK Biobank, everybody knows about, 7.5 million. EPIC, I believe, is close to 500,000 as well, etc. Some of those other ones that didn't make it are not part of the IHCC because in terms of that threshold of size that we've all agreed to, are less than 100K. So the Alliance study, for example, in Canada, that's a 30,000 participant size cohort. So it wouldn't necessarily be part of the IHCC. So this is a map. This map is embarrassing in terms of which countries and continents are represented here. Compared to the map that Jeff showed us earlier, we really want to improve this. So as you can see, we just had a lunch where we're talking about trying to get better inclusivity of LMIC countries. It's Nikki Mulder, who was meant to be speaking shortly, will be speaking tomorrow morning, who runs H3A BioNet, has just recently shared with us her data variables collected in the Pan-African program is there. So that will be one of our next jobs, is to try to include some of that work there. So the CHIPIT, I don't know why it's called, that's a typo. The CHIPIT program, this is what the CHIPIT program was that we started some years ago. So it included participants from cohorts of at least 50,000. So as I said, some of those cohorts won't make the grade going forward. But generally, this is a live catalog that you can it's already up. It's a portal where you can look up, say, blood pressure. And if blood pressure was captured in those 9 cohorts, see how harmonized that information is captured along those different cohorts as well. These are the cohort variables. I'm not going to I don't have enough time. I don't want to take too much time away from our speakers today. These are the ways we've kind of cataloged this information, taking information from the population cohorts, capturing information from the data dictionaries, and then asking what are the variables. And so these are, if you like, a list or a description of variables, and you can kind of get a handle from this slide as well as to how complete that information is for different cohorts. So I just picked cohorts where we've got information for ICD codes, and there's the information in terms of number of ICD codes for UK Biobank listed there in that histogram. So as I said, we're evaluating the harmonization potential. Jeff said he didn't want to use the word harmonization earlier, but in general, we need to get to that point at some point if we want to be doing genetic studies looking at phenotypes. We're pretty good with genetics in terms of integrating genomic data. To some extent, I almost think is easier than what we're trying to tackle here with some of this phenotypic information as well. So the portal is live. You can ask questions from the portal. You can ask, if you've got a cohort and you want to be picking variables. So I say, I'm only interested in, I don't know, setting up study inclusion criteria. And these are some exposures that you might be interested in, the outcomes, and then just pulling that information from that portal, from those cohorts. And this would be the result of a query from that portal as well. So I don't know if any of you recall, I hope some of you recall, an email went out some weeks ago. What we've tried to do is, as I said, capture what information we could without bothering you. and now we're trying to bother you with respect to getting data dictionary information. So we've sent an email. These are the people who've gotten back to us. Not enough of you have gotten back to us. So this is a great opportunity to remind you to please respond to my email. And you know, it's actually really fun. It's kind of fun for me, I mean, to look through these data dictionaries and see what people are capturing as well. So these are some of the folks who have already come back to us. And we're hearing from people like, we haven't yet built our data dictionary, and we will get that information back to you when we have it. And what we're hoping, and this could be discussed in the breakout sessions, is what is it that we could potentially provide back to you if you're a member or a friend of the IHC. I don't know what the participation nomenclature is here. Is this information? Would you like to see other people's data dictionaries, how they collated that information? How could that be useful for you as well? So these are the membership of IHCC Team A. I'm always first because I'm alphabetical, and that's the only reason why. But we've got some new folks who've been added to the program as well. And I think that's it. So as I said, I don't want to take too much time away from our speakers for this afternoon. So if there's time for a question, that'd be great. Otherwise, we can use it in the breakout session. Super. Okay. All right. So we're going to get. So Nikki, as I said, it won't be. Well, she'll be in the schedule for tomorrow morning. That's right. Okay. So our next presenter, we're going to keep the briefs, keep the introductions very brief. So Ellie Zaghini has kindly agreed to give us a presentation today on the work that she's doing in osteoarthritis, and so she's recently just moved to Munich, right? And glad to have you here.